Hello and welcome. Recombinant DNA technology, synonymous with gene cloning or genetic engineering, is really an amazing science which can literally turn the impossible into possible. It essentially involves isolating and loading of a piece of foreign DNA onto a man-made cloning vector, which in turn is inserted into a live host cell or an organism in vitro. This is done with a goal of creating novel genetic combinations that are of unmatched value and importance to research, medicine, agriculture, and industry. Recombinant DNA, abbreviated as rDNA, literally means a DNA construct that has been created by a combination of at least two DNA strands of mutually distinct origins. rDNA technology forms the most significant and powerful part of modern biotechnology, as it can integrate the DNA encoding any desired trait in any heterologous genome. This cutting and pasting of DNA has indeed proved to be useful in innumerable fields such as agriculture, medicinal and pharmaceutical products, environmental bioremediation, and whatnot. Recombinant DNA in a living organism was first accomplished in 1973 by Herbert Boyer of the University of California and Stanley Cohen of Stanford University, who demonstrated insertion of foreign DNA into plasmids using E. coli restriction enzymes. The basic tools of RDT are molecular scissors called restriction enzymes type 2 and molecular glue called DNA ligases, used to cut and paste respectively the DNA into the cloning vehicle, followed by insertion of this recombinant DNA into live cells, enabling them to acquire the desired trait whose DNA was initially selected. It was observed that bacterial cells do not take up foreign DNA readily. Therefore, they need to first be made so-called competent using calcium ions to accept the new DNA, followed by unique treatments such as a heat shock, electroporation, microinjection, biolistics, etc., which lets the foreign DNA in. In our present introductory video, we are providing a bird's eye view or an overview of RDT. The steps are: first, identification of the target gene to be integrated and expressed. Second, isolation of this gene by restriction endonuclease type 2. Third, selection of a suitable and compatible cloning vector, which can be plasmid or phage based. Fourth, inserting the gene into it using compatible restriction endonuclease and DNA ligase. Fifth, selecting the productive combinations and aiming to introduce these successfully into appropriate cells. Sixth, screening and selection of the transformed host cells from the mixture of cells using a selection pressure or a selectable marker. And the last, mass cultivation of these transformed live factories to indefinitely harvest our product of interest. Guys, it is really important to understand the framework of RDT approach. We shall be getting a simplified content elaborating each of these steps in our future short videos to make your concept crystal clear. Besides these, simple techniques of agarose gel electrophoresis for monitoring the cloning process at each stage by determining the size of the DNA, and polymerase chain reaction or PCR for amplifying DNA for various purposes during the cloning process are also of paramount importance and can't be overemphasized. Recombinant DNA technology is widely used in medicine, agriculture, and research. Although it has certainly revolutionized modern living, still it comes with some drawbacks as well. We shall discuss all of these in our forthcoming videos of this RDT series. Our endeavor is to enhance the grasp and interest of our esteemed audience in the field of RDT and also to make it very easy for you to follow and enjoy this branch of science. Keep watching and learning. We both are very grateful to you for your continued viewership and appreciation. Shall see you super soon. It's bye till next time.